Okay, well, everybody, welcome to our virtual brain maps. Thank you, Denis, for being here today. So, Denis Le Bihan is the founding director of NeuroSpin, an institute of the French CEA aimed at developing and using ultra-high field magnetic resonance up to 11.7 Tesla to understand the human brain. Dr. Le Bihan is especially credited with inventing and developing and introducing into research and clinical practice the concept of diffusion MRI, a powerful approach to study normal and diseased brain anatomy and function, as well as brain wiring. Le Bihan has received many awards, such as the Gold Medal of the International Society for Magnetic Resonance in Medicine, the Lounsbury Award for the National Academy of Sciences, and French Academy of Sciences, the Louis D. Award of the Institute of France, the Honda Prize and Louis Chantet Foundation Award. Dr. Le Bihan is a full member of the French Academy of Sciences and of the Academy of Technologies, and an associate member of the National Academy of Pharmacy and a corresponding member of the National Academy of Medicine. So with that, I'm gonna first give some comments on how to use tips during the use Zoom during this talk. So if you want to make a question, we will unmute you so you can ask your question by yourself. So in that case, use the questions and answer box. If you didn't sign up with your name, please write your name and we will unmute you. And if you just want us to read the question aloud and don't do it yourself, just write the whole question there. Please do not use the chat or do not raise your hands for that. Just use the question and answer box. So with that, Denis, I'm gonna let you go on with the talk, but we cannot see your screen right now, the presentation.
Okay, sorry, I just have some issue with PowerPoint. We disconnected, but
okay, so meanwhile, maybe I can do some other announcements. So we have moved our brain maps to Thursdays because on Wednesdays there is the radiology town hall. So to not overlap with those, we have moved to Wednesday. And also Gary is sending a weekly newsletter on Monday mornings, and in that email he will be announcing the videos that have been uploaded. I saw already on the chat that somebody requested for Denis' slides, but the talk will likely be available on YouTube in about a week from now, so wait for Gary's email and the announcement of this talk already on YouTube. If you are not receiving Gary's emails, just go to YouTube and subscribe to the BrainMap Martinos channel. So with that, Denis, thank you for being here with us today.
Okay, well, thank you very much for your kind invitation. I'm very happy to be with you. I don't know if I am in Boston or if you are all with me in my home, but anyway. So today I'll talk about diffusion fMRI. And let's start with some basic things you might know, of course, about BOLD fMRI. You know, BOLD is related to the neurovascular coupling between blood flow and activation. And this has been the basic principle for PET, using oxygen-15 water. And then you have to inject a radioactive tracer and use PET to see the activation. BOLD fMRI, which I think many of you are using now, which was set up especially by my friend Seiji Ogawa. And with BOLD fMRI, we are looking at brain activation through the increase in blood flow, which is also modulating the magnetization of water molecules through the ion present on the hemoglobin molecule. So with BOLD fMRI, we have thousands of papers which have been published looking at how the brain works in different conditions. So why do we need something better? Well, there are some catches that we have to consider when with BOLD fMRI. First, the mechanism underlying the increase in CEA in neurovascular coupling are not completely clear. Then, of course, if you are looking at the neurovascular coupling, you are not looking at neuron activation directly. And in fact, this is an issue because, as you know well, I think I have an issue with the PowerPoint. It is disconnecting all the time. I don't know why. I don't know why it's doing that. So, okay. So we might have some, it didn't do that before to me, but anyway. So because of that, the resolution in space and time of BOLD is somewhat limited. We are not looking at neurons directly. And we know also that sometimes the coupling may fail if in some diseases or during anesthesia, for instance. And just, this is a busy slide, but just look at on the left, some experiments which have been done in the cat visual cortex. If you look on the plot with the peaks in red and blue, blue is related to glutamate response according to the orientation of the stimuli, while red is related to the dilation of the small vessels. And as you see, the orientation dominance is lost when we look at, while it is still present, of course, with calcium and glutamate. So this is why diffusion MRI, which is not based on we'll try to convince you about that. But
Denise, can I interrupt you for a second? I think maybe you are touching your screen or moving a little bit the iPad, and so maybe it's interfering a little bit with the mic. So if people like...
I have to touch a screen to switch the slides.
Yeah, but like try to, I don't know, maybe try to touch in a place that is far away from the mic or something like that.
Okay, yeah, I see what you mean. Okay, I'll do my best, yeah. So with the diffusion fMRI, and hopefully what I believe is neuromechanical coupling, we are looking at microstructural events which are synchronized with neuronal activation, which could be more specific. So this is the outline of my talk. First, I'll give you a summary of what is diffusion MRI in case some of you are not familiar with what it is. Then I'll show you that we can do fMRI with diffusion instead of BOLD. Then I will try to convince you that the diffusion fMRI signal is not BOLD, it is not vascular. Then instead, I'll try to explain that we think that it is related to cell swelling and what kind of cells and so on. And finally, how cell swelling might decrease the ADC. So that's what I will talk about. So in case you are not familiar with diffusion MRI, let me quickly give you a summary. Diffusion MRI, as we know it today, was initiated in the mid-80s. So you see me here with a very old system in France. And the idea I had at that time is because images had a microscopic resolution, the idea was to see if we could get a microscopic resolution, like a virtual biopsy, without increasing physically the resolution, because we could not do that. So my idea was to use water molecules as spice, basically, because of a Brownian motion, and an article by Einstein showing that there is a relationship between the displacement of water molecules driven by diffusion. And for free water in the brain, at the temperature of the brain, it's a few microns, 50 milliseconds, which is basically the time we have with MRI to measure diffusion. So that's perfect. And this idea made it true because now we know that diffusion MRI has been used in many, many areas to look at the tissue microstructure. So water molecules can sense fibers, membranes, and so on, and give us some information about the microstructure of the tissue. Also, we cannot see it with our own eyes. So it's a trick, I would say, to be sensitive to the tissue microstructure, and more importantly in our case, to changes in the tissue microstructure. So how do we do it? We label space by what we call magnetic field gradients. And if molecules move a lot because of the diffusion process, the signal gets attenuated a lot, while if the diffusion is slow, and the movement is less and the attenuation of the signal is less. So this can be quantified using a very simple equation, in fact, which you have it, the exponential minus B ADC. I know now I can say it because many people ask me. The B comes from my name, but it was a joke at that time. But many people ask me, so I tell you. And then we published the first images of brain diffusion in 1985-86. This paper has been highly cited, so I'm extremely pleased. And diffusion MRI, as you know, has become a pillar of today's medical imaging. There are many applications. So the first one, I think, coming from your area, was on acute stroke in humans. And brain connectivity, so DTI, to make images of fiber connections, something I've done with Peter Basser. Our papers also have been highly cited. Oncology, to detect cancer, virtual elastography, and today I'll talk only about brain fMRI. So now I'll show you how we can detect neuronal activation with diffusion. But first, I'd like to show you something which is not diffusion MRI, but it could be a little bit confusing. It's about perfusion. So back in my paper in 1986, I showed that a diffusion MRI was also sensitive to perfusion. The idea was if blood moves in capillaries, which are almost randomly oriented in a tissue, they mimic a diffusion effect, so which we call pseudo diffusion, IVIM. And if you look at the plot on the left, you see the IVIM is basically a curvature yet you see at a very low B values. So this is sensitive to what's going on in the tissue. And this was controversial at the time. And in fact, when I was still a student, I tried to do fMRI with IVIM. So this is an image of the daughter of my mentor at the time. I was showing her some images and trying to see if the visual cortex could get increased perfusion. So again, neurovascular coupling, but with IVIM fMRI. So it worked a little bit, but it was not convincing at all. So I gave up using IVIM from fMRI. But there was something. And much later, Alan Song at Duke showed that IVIM indeed can be used to map activation of, for instance, here it is a motor finger tapping experiment. And the increase in perfusion that you have associated with the task can be seen with IVIM. But again, this is not at all what I will talk about today. So this is increase in ADC, so not diffusion. So it's very important. And we published a book last year about IVIM, where there is a chapter on this, especially on this particular issue, IVIM fMRI. And again, this is what you see at low B values. Now, if you look at the plot which just appeared on the right side, we see also that at the same time, there is a decrease in the ADC, the diffusion coefficient we measure, especially when we go to high B values. So there is a decreased diffusion, which happens during activation. And this is what we want to, that's what we want to talk about today. So I did a very preliminary experiment in France in 2001 that was published in PNAS. But the main experiment and the most convincing, to me at least, was performed when in Kyoto in 2006 with my colleagues. You see a photo there. And it worked very well. In fact, we could see that during a visual stimulation, the spatial localization for diffusion fMRIs was much more accurate than bold. But what was even more striking is the time course. This is a typical bold signal in the same subject. The black rectangle corresponds to the visual stimulation, and the blue curve is the bold response. Now, if we replace the bold sequence, which is basically a granular echo sequence, by a diffusion-weighted spin echo sequence, what we see is that during the visual stimulation, there is an increase in the signal. If you remember the slide, the second slide I showed you, or the third slide, increase in signal means decrease diffusion. So during activation, there is a decreased diffusion of water in the visual cortex. But if you look at the time course now, you see that the time course is very different. It starts much earlier. The peak is obtained right at the end of the stimulation window. And basically the overall response is a few seconds earlier than BOLD. So this, I mean, clearly, to us at least, was a proof that what we could see with diffusion was not BOLD. It was something else, something new, related to something going on in the tissue, so something of cellular or tissue origin. And if we look now at this paper by Schumers et al., where they look at the visual cortex of the ferret using 2-photon calcium imaging, you see on the right side, the response is about 5 seconds, so it's very similar to what we do with fMRI. The neuron and astrocyte, you see that there is a difference in time course. It is completely established today that astrocytes are responsible for the BOLD signal, while the red response for astrocytes fits very well with what we see with BOLD. But the neuronal response in blue, which is earlier, fits very well with what we see with diffusion MRI. So this was to us a good argument, indirect, of course, but that diffusion MRI, diffusion fMRI was something related to neuronal activation. And then the controversy started all over the place, including in your place, about maybe what we see is it's not tissue structure, but it could be some kind of disguised BOLD effect. I like this study by the group of Camille Hugerbill, where they checked in the cat visual cortex at 9.4 Tesla. And what they say, they see the same thing. They see a change in the ADC, so a decrease in ADC, which is occurring earlier than BOLD. It's not seen in the surface where you have big vessels, so there is a BOLD response there, but not a diffusion response. Everything looks good, right? And this is the time course they observe, so everything is in agreement. But this is what they write in the paper, which was a little bit shocking for me. So they say that it works, but we believe that it is not, it is a strong vascular contribution. They believe. So, I mean, science is not just believing, hopefully. So I was a little bit surprised of this statement. And basically, the idea was maybe what we see with diffusion has something to do with blood anyway, and not something with the tissue. And there was this paper by Carla Miller also, and with hypercapnia and blah, blah, blah. So several papers showed that maybe what we see is not maybe an artifact, but at least not related to tissue structure. So with Toshihiko Aso in Japan, we had a paper showing why we have this kind of disagreement. Depending on the weighting on diffusion, so the B value, you have two components. Like have very low B value, in fact, no B value at all, you have a spinnacle BOLD. When we increase the B value, the diffusion effect takes place and increases. So depending on the B value you use, you might switch from BOLD to diffusion. So that's very important, because if we want to do diffusion fMRI, we need to use high B values. But there is a very nice way to cancel any BOLD effect, is to calculate the ADC. So you take two B values and you calculate the ADC, then the residual BOLD, if there is a residual BOLD effect, will be canceled. So the conclusion of this first part is that the diffusion fMRI signal is not a filtered BOLD effect. Now I have to convince you why. So that's the second part of my talk. I give you some experimental results to show you why it is not a neurovascular coupling. So one first experiment we've done in the rats, somatosensory cortex with my colleagues from NeuroSpin. So it's a four-pole stimulation in the rat. And these are the LFPs that we have recorded in the cortex, layer 2 or 3. So you see clearly a demonstration that we have the activation. Now we compared diffusion fMRI and BOLD. So if you look at the images on the left, you see the activation with diffusion, you see the activation with the spinnacle BOLD, so basically B0, and we see also some responses with gradient echo.
Dennis? Excuse me,
yes.
Dennis, we have a question from Daniel Bradley. So Daniel, you wanna?
Yeah, okay.
Denis, hi. Real pleasure to speak with you. I've followed your literature for many years, and it's a real honor to see this. Thank you so much. So I had a question. Back in the kind of emerging days of BOLD, shortly after, there was some literature at Highfield where people were reporting, and maybe Ogawa was doing this as well, reporting a dip in the BOLD signal prior to the large increase in the BOLD signal.
Yeah.
How much further work was done exploring the origins?
If you could wait for the end of this slide, you'll get a response.
Oh, okay. I'll thank you, Denis.
You have already a hint there. So if you look on the left column, you see the activation spot for diffusion for spin echo BOLD, so B0, and the gradient echo. Now, if you give nitroprussate, something that Seiji Ogawa also did for BOLD, with nitroprussate, you increase the baseline blood flow, and then you kill the BOLD response. So what do we see? We see that the diffusion response is still there, right? The spin echo BOLD completely goes away, and with the gradient echo BOLD, we have a negative response. So this is a response to your question now. This is a time course with multiple stimulation. So you see that with nitroprussate, the B1000 and B2100 responses are still there, but the BOLD becomes negative now because what we see is a consumption of oxygen by the tissue. There is no increase in blood flow, but the oxygen is taken up. So the initial dip you are talking about is in fact related to this consumption. So before there is an increase in blood flow, I mean, at that time, the initial dip was interpreted as a mismatch between the consumption of oxygen, which starts quite early, and the real BOLD response, which is a little bit delayed. So I hope it is a response to your question. So we say it beautifully here because we kill the classical BOLD response. Is it okay?
That's perfect. Thanks so much, Denis.
Okay, thank you. So now, second experiment. I showed you already the ferret experiment. And now if you look at the same paper, they show something very interesting. They change the isoflurane concentration, so the degree of anesthesia in the animal. In the neurons, you have a small effect, which is of course related to the anesthesia, but for the astrocytes, you see a bigger effect. And from that, we expect, because astrocytes are related to the neurovascular coupling, a bigger effect for BOLD. So we did the experiment again in rats. So it's a resting state. We just look at the effect of anesthesia. So we switch from 1.5 to 2 and 2.5%. And we use another drug which has less effect on blood flow, which is medetomidine. And look at the results. It is really clear. I mean, if we look now at a comparison between 1.5 and 2.5% for isoflurane, you see for BOLD, you have activation almost everywhere. It's not activation. You have increased BOLD response almost everywhere. If you look now at the same effect for the ADC, you see that the effect is much smaller, much more localized, but there is an effect. Now, if you switch to medetomidine, for BOLD, you get a negative response, which is quite surprising. While for the ADC, you still get some positive changes. So the ADC is changing in the same way, but the effect is smaller. So let's look at the quantitative results now. So the ADC goes up with the concentration of anesthetic agent, whatever the brand. So isoflurane or medetomidine, if we increase the concentration, there is more anesthesia and the ADC goes up, which is expected because we've seen that the ADC goes down during activation. But for BOLD, you see that for isoflurane is going up and for medetomidine is going down, which is a little bit scary because then it has nothing to do with neuronal activation or neural function. And in fact, the BOLD response is completely parallel with the blood pressure that the trend you have with isoflurane and medetomidine. So diffusion is completely immune of those changes in blood pressure. And if you look at the time course now, you see also that for the ADC, it's perfectly straight, perfectly stable in time, while for BOLD, there is a drift. So again, these experiments I think, is a clear demonstration that we can see the neuron deactivation due to anesthesia independently of any vascular effects. So the vascular effects are huge here, and if we use BOLD to assess resting state, for instance, we have to be careful about this pitfall. I don't want to say it's not possible, just that it is a confound. Now, I will show you some results which have been published recently by the group of Noam Shemash, and which are extremely impressive, I have to say. First, they look at the layer-specific connectivity in the thalamocortical pathway. So it's, again, the same. We'll use this experimental setup quite a lot during my talk. It's a 4-pole stimulation again. And basically, the circuit goes to the thalamus and to the sensory cortex. So what is known from the literature is that the activation of the sensory cortex is not very local. So you get activation of the whole sensomotory cortex in the area corresponding to the 4-pole. And in the thalamus, most often, you don't see any response. So first, what is very important to say is that in their case, they don't use any model. They look at the distribution of the signals in regions of the brain. They don't make any assumptions about the responses, the shape of the responses, anything. It's very plain. And what they observe, for instance, in the sensory cortex S1, there is increased signal for BOLD and an increased signal with diffusion fMRI, which we expect, of course. Now, if we look at the thalamus, you see that it could obtain diffusion fMRI response, but nothing with BOLD, as was earlier published in the literature. Now, what they've done here is they look at the correlation with the frequency. So basically, a spectrum analysis, and you have the paradigm in A, and then you have the frequency. So basically, the first order and the second order are the main components. So on the top, it's a typical example for a single subject, and then you have the average. I think there are five animals. So what you see in E on the left is diffusion fMRI, where you see that the response is really not the same within the layers of the cortex. While for BOLD, you have all the sensory cortex, which is lighting up. Now, if you look at the thalamus, on the diffusion fMRI, so that's F, you see there is a response that you can clearly identify, while with BOLD, there's absolutely no response. The next thing they've done was to do a time coherence between the sensomotory cortex, S1, and VPL. So they tried to see if there was any time relationship between the activation time course. So in C, you can see that if we take the sensory cortex as a seed, you can detect a high correlation with the VPL in the thalamus, while with BOLD, you don't see anything. And again, in E, if you take now the opposite, you take a seed in the thalamus, you see that there is a correlation with something in the sensory cortex, S1, while with BOLD, there is nothing. So, yes?
Excuse me, we have another question from Michael Bernier.
Yes?
Michael, do you want to make your question?
Yes, sorry, just related to one of the experiments you show, I think 2 slides before. I was just wondering, like the negative BOLD response that you observe, could it not be related to changes in cerebral blood volume instead of like CMRO2 or perfusion or something like this?
It could, I agree, it could, but based on, especially Seiji Ogawa did the very same experiment to demonstrate BOLD, that BOLD was related to the neurovascular coupling, and the conclusion was it was an increase in oxygen consumption. But I agree, I agree that it could be also a change in the vascular volume and not blood flow. Is that what you mean? Yeah.
Yes, that's it.
Yeah. No, it is possible. It's not really what I wanted to show. I'm focusing on diffusion, but you are right. It's one interpretation which you find in the literature, but you cannot exclude it is a change in the blood volume. But in any case, you see what for diffusion, it doesn't change under natural appreciate. That was the main conclusion of this slide, is that if you increase the blood flow as a baseline and you cannot change it upon activation, it has no effect on the diffusion response, but a very deep, it has a deep impact on the ball response. That was the message of this slide. Okay.
Okay. Thank you very much.
You're welcome. So to go back to this very cute experiment or study, several experiments. So first, they show that the diffusion fMRI response is specific to layers 4 and 5. They show that diffusion we can detect VPL activation, but not with BOLD. And there is a temporal coherence between VPL and S1 signals in layer 4. Now, something else from the same group, where they try now to have a more detailed response in terms of temporal feature. So to do that, we are going to the same setup again, the 4-pole stimulation, but this time they use a line acquisition. So instead of a slice, you will look at the activation along colon, if you like. And this is also extremely beautiful. I mean, I'm very impressed by what they've done. If you look at A, C, and E, and the curves which are on the right, so the green rectangle corresponds to the stimulation. So you see with BOLD, the response is delayed, which we know, of course, but the diffusion fMRI response is much earlier. If you look, in fact, at the bottom of C, you see that it starts almost less than 100 milliseconds after the onset of the stimulation. And then it propagates along the layers, and there's clearly an influx of activation along the cortex, and there is also a response after the green rectangle. Looking at the ADC now, you see that the ADC is completely locked with the activation and occurs in also very specific regions. So to summarize this result, so in BOLD, so for the question of initial dip, you see it here a little bit. If you look at B, you see that there is a small decrease at the beginning. So that is supposed to be the initial dip, but then the peak occurs about 1.3 seconds after the neuronal response, and it is global. The diffusion-weighted signal is much more related to the stimulus. It is locked with the neuronal response, especially the ADC. But there is an overshoot that is present, and something which has to be investigated also, and I will show you some hints about that later. Now, they also compared the diffusion response and the BOLD response with some optical signals they have obtained in a model of acute hippocampal and cortical slices. And they use optogenetic stimulation. So at least they know very accurately the time course of the activation. So I don't have to go to the details, but if you look at D, you see that there is an optical response which is very accurate in space and time, and I'll show you later, but the iOS is now really clearly interpreted in terms of cell swelling, but that's not the goal of this slide. Just to show at the time course now in E, in black, you have the optical response, in blue, the diffusion fMRI response, and you see that during the stimulation, they match perfectly. But the diffusion response continues a little bit more after the end of the stimulation. If you look at the ADC now, it is completely linked to the optical change, while the BOLD response, as we know, has an initial dip and this peak, which is a little bit later. So I hope you would agree that those results are quite convincing that BOLD and diffusion are looking at different things. So the last experiment for this part is another optogenetic activation experiment, where again they compared BOLD and diffusion, and they use optogenetic stimulation. But one pitfall, of course, if you do optogenetic stimulation, is you are using light, and light could be hitting the tissue. So what they've done is to compare real stimulation and just light to see, so at a different frequency, to compare the effect of heating and a neuronal response. So let me, because I cannot show you the plot very accurately, but what you have to see, if you look, for instance, at A and B, and so the GE bold and the B500, and then on C and D, it's still diffusion, but with B1000 and 1500. So what we have to see in those plots is that for the optogenetic stimulation, you get, so it's a blue curve for bold and the blue curve for diffusion, you get a response, which we expect. Now, for the light application, so no stimulation, we see in bold, it could be a negative response, red, or a positive response in green. For diffusion, you have always a negative response in red. The reason why, yes?
Sorry, Denis, sorry to interrupt you. So Bruce Rosen is wanting to make a question. Bruce, can you talk?
Hi, Denis. Thank you very much for your willingness to do this. Fantastic discussion. On your previous slide, just very quickly, you showed this very compelling data of the match of the time course of the intrinsic optical signal with the diffusion. But what specifically was that optical recording? What were they measuring?
They were measuring the refringence of the optical signal. I cannot, unfortunately, I'm not sure I can go back without, maybe I can. Yeah, okay. Unfortunately, because I had no time, I didn't prepare the setup.
Sure. Yeah, I was just curious.
I have a slide later to show what, I'm sorry?
I was just curious what exactly that optical measurement was. Obviously, there are lots of different optical measurements.
Yes, but I have a slide later specifically on iOS. So maybe you will get a response there. They are measuring a change in the optical property of the tissue, right?
I'll wait for the.
Okay, I'll show you. It is related now, very well established to cell swelling, right? So when you stimulate, here they use again optogenetic stimulation. You have a change in the tissue structure, which is changing the way the light is diffusing in the tissue. So that's what they see with the red, with the black curve. It is
I guess kind of like the MR though, a change in an optical signal, can be interpreted probably in a number of ways. So the kind of
not here, because there is no blood flow. There is no blood flow. It's a slice. It's not in. It is ex vivo. So,
no, I get that. My only point is that a change in an optical signal also could have a number of biophysical.
Absolutely, but let's wait for the
that sounds good.
Another slide specifically dedicated. Well, not completely dedicated, but you have more details on that, right?
That sounds great.
Okay, so let's go back to this slide. So the point here is that if you look at E and F now, you see that by increasing the B value for the light application, the signal gets deeper and deeper because we have an increase in the ADC. The tissue is heating, diffusion is faster. With the optogenetic stimulation, we have exactly the opposite. The signal is increasing more at high B values and low B value because we have a decrease in the ADC. Also, I'd like to point out that if you look at the amplitude of the effect for bold and diffusion, you see that the diffusion response is larger than bold. I often hear that diffusion fMRI is difficult because the amplitude of the response is smaller than bold. That's absolutely not true. In our hands, at least, this is not our work, but it is very similar. The diffusion response in amplitude is at least the same, if not larger than bold. The issue is SNR, which is something we can discuss later. Anyway, so if people are considering using optogenetic stimulation, I think a concern is heating. We have to be extremely careful, especially with bold, because the response could go either way, up or down, depending on the proximity of the voxels you are looking at with the tip of the laser fiber. While for diffusion, it's very clear. If you have heating, you have increased diffusion. if you have decreased diffusion, it's not eating. Of course, there could be a balance. It could have some heating in addition, but it is very clear-cut. So I hope now you are convinced that the diffusion fMRI signal is not bald, it's not vascular. So now let's go to see that it is related to cell swelling and what kind of cell swelling. So first, in general, if you are not familiar, the idea that there is some mechanical response when neurons get activated is not recent. There are very old papers from the 60s and 70s. Many are from Japanese groups. I'll just show you an example here. Again, maybe Bruce that will respond to your question. And with optical imaging and infrared transmitters, they also look at different regions in the hippocampus, and they see a very sharp response, which has been interpreted in terms of cell swelling, even in those very early papers. Tasaki did something which is also quite interesting. He looked at bumps produced by action potentials when they are propagating along fibers. So using a piezoelectric sensor, you can detect a bump in the membrane when the action potentials goes at this specific spot. Now, something more global, it's extra physiological, but there are all papers showing that when you have cell swelling, for instance, induced by status epilepticus, spreading depression, and so on, we have a decrease in ADC, which is also related to the cell swelling. And as a third evidence, indirect, I agree, but this work by Flint, where they measured also in a slice, a hippocampal slice, activation by kainate. So they see, visually, they see the swelling, so that's clear, and they observe a decreased ADC. Now, when they block the kainate receptors, they see that there is no longer any swelling and no change in the ADC. If they decrease as a baseline using an NMDA receptor antagonist, then the response gets bigger, but the swelling gets bigger and the ADC decrease gets also bigger. So all these facts, I think, suggest very highly that there is something related to cell swelling. So we did our own experiment recently with Dr. Abe. So we, in this experiment, we have anesthetized rats with medetomidine, which we know now is safer in terms of bald and diffusion. And the goal was to wake up the animal, as you can see on the small movie, by stimulating the thalamus central median nucleus. So the animal is anesthetized, but if you stimulate electrically this nucleus, the animal will wake up, right? So you've seen the movie, I'll show it again. Okay, so what we've done now is at the same spot to inject either furosemide, which is a swelling inhibitor, or I would not say water because water is toxic. So we used a decreased solution, diluted solution of CSF, which is a swelling inducer. And what we observe, of course, is a change in the ADC. Under furosemide, the ADC goes up, and under this solution, H80, the ADC goes down. So, which is linked to the swelling or the blocking of swelling. Now, what we've done here is to look at the threshold to wake up the animal. So, if you look at the plot on the top or the right, we give a special current, a specific level of current to wake up the animal, and under furosemide, there is no more, the animal cannot be awakened anymore at this threshold. So basically something has changed. The furosemide, which has blocked swelling, has also blocked the possibility to wake up the animal. So what we've done is to adjust the threshold. And we did that for the two experiments using furosemide and using H80. So what we see is that, so the plot in the middle, when we inhibit cell swelling, we increase the threshold necessary to wake up the animal. when we induce cell swelling, we decrease the threshold to wake up the animal. So again, this is, to us, a very strong indication that by the swelling is not just there for us to do fMRI. The swelling has to do with how the brain works. Basically, if you prevent swelling, the animal cannot wake up. So it has a big effect on the neuronal system. So it's just that diffusion fMRI is just something to reveal what's going on in the brain. So it's not neurovascular coupling, it's something else. And basically, it is related to what's going on in the synapses. So let's look at this other experiment, not an experiment, just some other data we've done here. So again, the animal is now resting, no activation. And we tried to look in the brain where, when we change the ADC in the central median nucleus, there is also a change in the ADC. And what we could see here is that there are regions which are connected to the CM, especially the somatosensory cortex and cingulate cortex, but other regions as well. So if you manipulate the ADC in CM, just manipulate the ADC, you also change the ADC remotely in regions which are connected nearly to the CM. So basically, this would suggest that if you manipulate the, if you inject furosemide, you manipulate the activity level in this area of the brain, CM. But by doing that, you also act on the activity level in remote areas, which can be detected by changing in the ADC. Of course, there is no diffusion of furosemide in those regions. Okay, it's a very, very focal injection. So to summarize those results, and this point of my talk, we believe, at least I believe, but I think I'm not the only one now, that what we see with functional diffusion MRI is something related to neuronal swelling and synaptic activity. So, I mean, the synapses that we have in the voxel, I mean, the number is really, really, really high. This is a movie here, which I've got from a group from Osaka, where you see the dendritic spines. Everything is swelling all the time, swelling, shrinking. It's a continuous dynamic effect. So this is supporting, in fact, the activation in the tissue. So look at the synaptic density. It's 800 millions per millimeters cube, and the dendritic density is extremely high. So if I quote now Cahal, our pioneer, it says that neurons are the mysterious butterflies of the soul, which could one day reveal the secrets of our mortal life. Okay, but next sentence is, I love it. The state of activity would correspond to the swelling and elongation of the dendritic spines, and the resting state, sleep or inactivity, to the retraction, 1899. So I have not invented anything, right? It's there already. It's just that it has to be shown. And I think diffusion fMRI is one way to show it. So this is a cartoon just to see the message I'd like to carry here is that if you take a resting state, there is a dynamic. So there are some neurons which get activated, some are resting. So you have some swelling, which is in a baseline. When you activate this part of the cortex, there is more swelling occurring, but at the dendritic spine level, and water diffusion goes down. Now, during anesthesia, there is less activity going on, so the global amount of swelling is decreasing, and water diffusion goes up. So now I'll show you something else, just a parenthesis. Now this is part of a more global picture. I have a paper which is on the way, using the relativity theory to explain how information processing can propagate into the brain, but it's not ready yet. Maybe that will be the object of another lecture. Okay, so now I'll show you some experiments which are not fMRI, which are not MRI at all, in fact, to show you that this idea that there are changes in the mechanical changes in dendrites is not just a crazy idea. I showed you that there were papers very early on, but these are very recent papers. So this is maybe to help you addressing your question, Bruce. So in the first paper, I will not go to the details because I can see time is running out, but you can look at the references. I put everything there. So they use polarized light imaging. So it's not just a transmittance iOS, which is, in fact, I mean, if you look at all these papers, I agree there are several mechanisms possible, but most of the time you see that it is neuronal swelling. But now they use a birefringence, which means that they use polarized light, which is a little more accurate. And what they conclude is, from what they see, again, if you look at C, for instance, the amplitude of the response, and D and E, so for the transmittance, which is the same as I showed earlier, but the birefringence now, which is something more specific, what they say is that the signal response is associated with synaptic transmission in brain slices, and they say also it's related to post-synaptic cell swelling. So these are very accurate post-synaptic cell swelling, and they also mention the water influx to the membranes. And this occurs in the spines, where they can see a change in shape, volume, and number. Another paper, which is also cute, where they look at the hippocampal slices, and they look at the time course also with optical imaging, iOS. And if you see a B and C, you see that there is a response, an early response at 50 to 90 milliseconds, and then a later response. In fact, those responses are also related to swelling, but of neurons for the early response and of astrocytes for the later response. And this might explain what I showed you at the beginning, that there is an early response for diffusion, but there could be also a late response, and the late response could be linked to astrocyte. Again, I'm very quick here, I know, because time is running out, but you will have the slides, so you can look later. Now, this is another paper where they describe, they see a mechanical spike, which is synchronized with action potentials. So they claim that there is water movement, which is responsible for, if you look at the plot, DZ is a change in the position of the membrane. And so there is something there. And the final example, there are many papers, but I showed you the most appealing ones. This is a simulation, but they showed how mechanical waves could be associated with action potential propagation. So on the plot on the right, you see the voltage, and you see the membrane displacement. And there are two kinds of displacement: perpendicular to the surface to the membrane, and parallel to the membrane. So again, I just invite you to read all those papers. I agree that there is discussion on the mechanism and everything. Everything is converging, I think, very well. Okay, last part now. How cell swelling may decrease the ADC. This one is a little more speculative, or you might say everything is speculative, but I hope I convinced you a little bit today, but this one is more speculative than everything else. So there are also some early papers, as I said, which show that when there is cell swelling, there is a decrease in ADC. First, it is what we see in acute stroke during cytotoxic edema. If you change the osmolality, you make cell swells, the ADC goes down. And I already showed you this slide about spreading depression. Okay, so what we've done is to look really at tissue in at high resolution using 7.2 system, a 7.2 MRI system, and we look at our friend, the Aplasia californica. We are cheating a little bit because neurons are a little bit bigger than in mammals. So it is a little bit easier, but we can look at individual neurons and we can look at ganglia. So first we tried with a hypotonic shock that was worked by my former student, PhD student. What we see is that during the hypotonic shock, inside the neurons, there is an increased diffusion. This is in fact what we expect, because that means we dilute the content of the body of the neuron, and the ADC should go down. But as I said, what we observe during swelling is a decreased diffusion. So if you look on the right, at the ganglia level now, if you take a region interest, taking all the neurons, we see a decrease in diffusion. So we have repeated the experiment now with Wabine stimulation, and we get, which is more physiological than hypotonic shock, and we get exactly the same response. So inside neurons, ADC goes up, dilution, but outside, if we take the outside and inside, so at ganglia level, we see a decrease in diffusion. So what's the difference between inside neurons and ganglion? I mean, it's just extracellular space in membranes. We did all the calculation to see, because the extracellular space is quite big in those preparations. It's not like in the cortex. So the extracellular space, changes in extracellular space cannot explain the difference. So this is how I came up with this. And I forgot to say that we have another paper where we showed if we use dopamine now, which is even more physiological, we have exactly the same level, the same response, increased diffusion inside neurons and decreased diffusion at the ganglia level. So the model which I'm trying to pursue, again, it is highly speculative, I completely agree, is that we are not looking at intra or extracellular water, but at a layer of water which is bound to the cellular membrane. So that's what I call a slow diffusion pool. And water molecules are dynamically exchanging between bulk water and this pool. So any change, any modulation of the surface of the cells will have an effect on diffusion. If you have cell swelling, you increase the quantity of this bound water, so you have decreased diffusion, and you have shrinking of the cells, you have the exactly opposite effect. So we've done some experiments to go along this line using digital holography microscopy. So the setup is on the right, on the left, but I have no time. So we can look at with light at a single neuron, and we can measure also the refractory index, which is telling us something about the structure of the water in the neuron. So the equation is there, but don't pay attention to that. So when we stimulate the neuron, there are two things which are occurring. First, we see with our own eyes, there is a swelling, so it's not an illusion, there is swelling. But also, we see that the refractory index changes, and a lot, in fact, which means that there are probably changes in the structure of the water. But we can also measure, so we can evaluate using the equation which is there, the change in link the refractory index, refractive index that we measure with the quantity of water molecules which have gone inside the cell. But at the same time, because we have the voltage, we know how many ions have crossed the membrane. Usually people assume that the water which goes with ions is just solvation, so 6 to 8 ions per, 6 to 8 water molecules per ion. But if we make the calculation, we find that there are between 300 and 600 water molecules per ion. So clearly this water flux has nothing to do with the flux of ion. It is something else. Just to point out, I will not give you any response. It's just to tell you that in order for cells to swell at the level we see, there must be many water molecules to cross the membrane. And the cytoskeleton also has to play a role. Just to give you a hint of the power of water. Look, I'm sure you have experience on the left, these towels that people give you at the restaurant to wash your hand. You put a drop of water and poof, in a few seconds you have something big. Now, if you have a garden, especially in Japan, if you have bamboos, you see that the bamboos can even go inside your house, and they are very powerful. But it's just because of the water molecules. So the water structure is something that is of very high interest, at least to me. And like this quote by water is H2O, hydrogen 2 parts, oxygen 1. But there is also a third thing that makes it water, and nobody knows what it is. So, of course, the movie, when I saw the shape of water, I immediately went to the movie to see, but in fact, there is no clue in the movie, sorry. So basically, I think that we have to consider water as not a single, H2O doesn't exist, in fact. It's a social water, it's a balance, it's an equilibrium. And some papers, I'm not a specialist, I have to say, but some papers are really suggesting that on the localization in space of the water networks, there are variations in the diffusion of the proton and finally of the water molecules, depending on the quantity of defects. So this is the cartoon, but it is explaining what people have observed. If you have polarized layers, and membranes, cell membranes are polarized layers, you get something in the structure of water, which is different depending on the distance from the membrane. So close to the membrane, water is sticking basically to the charges, and you have slow diffusion and high viscosity. Now, the big controversy is how far is this layer extending? And then you have basically two schools, what I call hardcore physicists, who consider that if you consider only the electrostatic charges, it's very small, and the temporal coherence is extremely small, so it cannot explain what we see. But there are also papers showing that even on not only biological cells, but all other objects, that this effect could extend to 180 nanometers. So you see, I don't know, but it could well explain, in fact, what we see. So there is really water cytoskeleton interacting next to the membrane. So to understand better, I edited a book on water, and I learned a lot, and I suggest you to read this book. It's not easy to read, I have to say, but there are many experiments showing that we know almost nothing about water. That's the conclusion. So to summarize, because I think it's time, first, I think I showed you that water diffusion goes down during brain activation, and the response is faster than bold, which points out to something new. Second line of experiment, we have shown that the diffusion fMRI signal, when you use high B values, and even more if you use the ADC, is not related to something vascular. It's something else occurring in tissues. Third conclusion, when we manipulate the amount of swelling in the neuronal tissue, we manipulate not only the diffusion signal, but we manipulate the response of the neuronal tissue. So synaptic activity is related, is involved with the swelling, and maybe that's what at least I propose. Diffusion fMRI is linked to the swelling. And this swelling, which leads to a decreased ADC at tissue level, might result from this model of slow diffusing layer next to the membrane. So the conclusion would be that if you want to do neuropsychology, especially normal subjects, bold fMRI is perfect. It's easy, you get whatever you like, it's perfect. There are thousands of papers. But if you are more interested in something which I call, well, don't quote me on neuropsychology, it's not neuroscience, okay? But what I mean here is that if you want to do something more specific, where you require higher resolution or higher temporal spatial resolution, more neural specificity, or connectivity, even in resting state, especially if you do preclinical studies with anesthesia, I highly recommend you use diffusion fMRI. Now, if you allow me, I have only one more slide. Is it okay or not?
Yes, of course.
Yeah, okay, well, I'm not sure. So last slide is very, very speculative, but it's a lot of fun. Now, I'm just talking about water, okay? What is diffusion fMRI experiments mean for me? Well, first, diffusion fMRI is just a tool. What is interesting is that if neuronal swelling occurs, as I said, there is a reason. So water has some kind of role in neuronal function, action potential, depolarization. And especially mechanically, it has to do with, if it is swelling, with the cytoskeleton interaction and the dendritic spines. Second point is, oops, I don't know why I have this. Maybe because I'm talking too long and automatic switch. Okay, it's the last slide, so it's easy. Okay, I'm happy it happened only at the end. I was afraid. Okay, so second slide, very provocative. Why do we have increased CDF, neurovascular coupling? We know that there is a lot of oxygen in glucose in the blood in the baseline condition. So why do we need more? And if you look on the right, the cartoon of Egyptians, they were not keeping the brain in the mummies because they considered the brain was just a radiator to evacuate the heat. It had nothing important to do. So the provocative idea, but I'm not the only one to have it, is that the increase in blood flow, in fact, is related to the fact that we have to evacuate the heat in tissues which are activated. In fact, if you change the water order, you produce heat, and this heat is toxic. So that's why, to tease Seiji Ogawa, sometimes I said, you should have called your method not ball fMRI, but cold fMRI. So, but some people have measured the temperature increase. So the same guy who could see the bump in the membrane of the axons have measured also the temperature increase, 23 microdegree. But if you consider the number of axons or dendrites you have in a neural tissue, it could be up to 1 degree. So the increase in cerebral blood flow might be just firemen coming to cool down the area which has been activated and also to restore the energy, but that takes time. And that's why the ball response is not accurate in time. And that's why the ball response is not very localized, because it doesn't matter if you have to cool an area of the brain, there is no emergency and you don't have to be very accurate in space. And the last point is that this electromechanical coupling might have a very important role in how information is transferred between neurons. So the dogma is that we have synaptic transmission. But what if neurons could be considered as piezoelectrical transducers? So as I've seen, that if there is activation, you change the shape of the neuron, but it goes the other way also. If you change the shape of the neuron, if you change the size of dendrite, you might induce activation. So if most of the time we consider the green slide with neurons which are very far apart, but that's not the truth. As you know, the tissue is very compact. All neurons, dendrites, and everything is very close, very touching. No social distancing in the brain. So when one cell or one part of the cell gets swelling, it has an impact immediately on the cells which are nearby. Sometimes, as a joke, I said, if you are in a crowded Tokyo metro, so in these days it's not the case anymore, but if somebody is sneezing, the sneeze is transmitted immediately to the proximity just because of the mechanical movement. And this has been, sorry for that, well, if you look at the bottom, it has been proposed as a mechanism to explain why we can get activation in the cortex using a transcranial focused ultrasounds as a way to stimulate. So not using electrodes, because this now can be done in the human brain, but to have mechanical stimulation of the cortex, so mechanical effects, which means to be electromechanical coupling. So again, this slide is highly provocative on purpose, just to let you dream of some other experiments or whatever you want to do. So thank you for your attention. I'm sorry I have, my time is largely over, but I'm still here. If you have questions, of course, I would be very happy to respond.
Okay, great. Thank you so much for this talk. We have a question from Ken. Give me a second. Ken, I unmuted you. You can ask your question.
Ken, we can't hear you. You actually might have to unmute yourself manually now that you've been kind of given permission to speak. That's what I had to do in any case.
Can you hear me now?
Yes, yes.
Oh, I was saying that if heat increases during activation, wouldn't it lead to a temperature artifact of diffusion signal? Now you would have an increase of ADC instead of decrease of ADC. Would that be a consideration?
Of course, but there is no increase in temperature because you have the bald effect.
So that would suggest that you would get this flip in the diffusion signal if you say did your nitroprusside experiment. In other words, produced a constant flow in the face of any changes in activation.
Right.
Imagine, what would you see in that case?
Yes, that's right. But on the slides I showed you, we don't see something like that, which means that if there is a temperature increase, it might be toxic. It might be there, but not big enough to change the ADC. It's not like the optogenetic stimulation where you use light and then you put a lot of energy locally, then you have really an increased temperature. But it could be that the increase in temperature is not enough to drive a diffusion increase, a diffusion coefficient increase, but still enough to be toxic for neurons. Again, it's a very provocative hypothesis. I'm not the first one to have suggested it, but the addition here is that, you know, if you have a lake and you make ice, you have to, there is a release of heat. And you know that in California, when they have oranges and when in the winter they are afraid that the oranges might freeze, they put water on the ground, and when the water freezes, it is releasing heat which protects the oranges. So what I am adding here is that if there is this increase in water ordering, that could release more heat, and so that's maybe the reason why there is a need to increase the blood flow. But again, it's just a crazy idea. I mean, that's not the main point of my talk. I hope you understand that.
Yes, it's an interesting idea. Yeah.
Any other questions?
Denis, I was going to wait for others, but I can't help but ask another question. It's super talk, by the way. Very, very interesting and very comprehensive in reviewing the literature on this. One of the things that you know, we, of course, many others have been thinking about recently is using you know, other diffusion measures to try to get a better sense of spatial scale within tissues, you know, microstructure measurements. Given some of the ideas you've got on the origins, could you imagine kind of classes of diffusion-related experiments that might allow us to kind of pin down within at least the spatial scale of the tissues that we think, or tissue subsections, cellular sections that we think might be most likely to be involved in these kind of bulk diffusion changes that you're able to demonstrate here? Is there some role to play for some of these other microstructure-oriented diffusion methodologies to help address this question?
Yes, in fact, I'm very jealous because you have such powerful gradients, which we don't have. And with a connectome gradient, and I've heard that you will get even higher gradient power, you are able to very specifically address compartments. So when I showed you the 17 Tesla experiment, and we have a 1 Tesla per meter coil for the gradient. We can go to 1 Tesla per meter, so it's huge, and we could differentiate signals coming from inside neurons and outside neurons because of the size. But now, if you can do that in humans by modulating the diffusion time, and the b-value, you might be able to differentiate the global signal, intra-dendrite and outside dendrites, and signals coming only from axons and dendrites. And by comparing the two, you might have some clues about what's going on. You might see that inside the axons, the ADC goes up, but if you now take the average, you get the decrease in the ADC. So basically exactly what we found with the aplasia californica. But if you could do it in humans, of course, it is a completely different story.
Well, maybe that's a conversation we should continue and think about collaborating on.
Yes.
So Robert Barry has a question. Roy, can you talk? You have to unmute yourself.
All right, can you hear me?
Yes.
Yep.
So thank you very much for your talk. Just a very high level question. In your opinion, from everything you've seen over the last couple decades, what's like the main thing that people get wrong in either the processing and or interpretation of their diffusion MRI data?
Tricky question.
Completely open-ended.
Well, first, I would say that you need good SNR, and if you don't have a stable gradients, then the SNR is not good. As I said, the diffusion response, the amplitude is bigger than BOLD very often, but the noise is also bigger than what you get with the gradient echo sequence at short echo time. The big problem is that in order to put the gradients and get high B values, you need long echo times, and this is killing us in terms of SNR. So in your place, because you are so lucky to have this wonderful toy, the gradient connectome system, you may get shorter TEs and boost the SNR. And I think that's the main limitation. Why people are afraid of doing diffusion MRI, fMRIs, they think that the results will not be as strong as BOLD, and which is not completely wrong in terms of what I'm saying. So that's the first catch. The second problem is if you publish a paper, most people I have seen as a reviewer when I'm reviewing other articles, what the other reviewers say, or my own papers, what I get. If you only do diffusion-weighted imaging, so you don't show the ADC, you always have the critics that, well, but you know, you have T2 sensitivity, so you have BOLD, we don't know if it is BOLD or diffusion, blah, blah, blah. So it is very important to go to the ADC and really to keep the discussion short. I would say, don't use 0 and 1000. Just go to higher, the lowest B value, you should use 200 or 400. So you are away from IVIM, you are away from everything. And then for the highest B value, in order to get the highest sensitivity is a trade-off between SNR and diffusion sensitivity. In my first paper, we went up to 2,400, but now we backed up to 1,800. But again, with your gradients, you might be able to go to higher B values. So I think that's the main two issues. In terms of processing, there's nothing difficult. You can use whatever tool you have with BOLD. I mean, once you have a time to, I mean, doesn't care about what the origin of the signal. But these are the main limitations: to get enough SNR and use the ADC, and the ADC calculated from somewhat higher B values that are used for clinical applications.
Denis, just a quick follow-up. Just looking, say, even at this last figure and looking at the amplitude, which are normalized here in the respective error bars, it looks like the kind of contrast to noise is maybe a factor of 3 different than BOLD, or 2. What's your kind of estimate, again, using kind of conventional B values like you've used here, and roughly 2.
Yeah, I think the figure is a little bit misleading, because the number of averages is not the same for BOLD and diffusion.
Okay,
so,
fair enough.
Yeah, yes.
What's the difference?
That's a very good point for the question. You need, most of the time, because of this SNR issue, you need to repeat the activation and average out several runs, several blocks of activation. Sometimes with BOLD now you can do a single activation. With diffusion, it is almost it is very difficult. So you need to average out, let's say, 2 or 3 or 5 runs. That's a good point. Yeah. Okay.
Great. Thanks.
All right. So if there are no other questions, yeah, we have another one. So we heard it from an anonymous person. In the result from Noam Shemesh experiments, why does this spin echo BOLD signal vanish when the diffusion weighting is added? The ADC may remove the BOLD contribution, but why is BOLD removed from the diffusion weighted signal?
I'm not sure I understood the beginning of the question.
So in the result, yeah.
Could you repeat the question again? I could not catch the beginning. Sorry.
Yeah. In the result from Noam Shemesh experiments, why does why does the spin echo ball signal vanish when the diffusion weighted is added?
I mean, the ball response doesn't vanish. I mean, you have a T2, the T2, it is a spin echo sequence, so you have T2 effect, okay? And because we use EPI, you even have a small T2 star effect. So whatever the B value, you have a small residual T2 star effect, which is bold. But in addition, when you increase the B value, you build up a diffusion sensitivity. So the higher the B value, the higher diffusion component there is to the signal. But you have always diffusion and a little bit of bold. To remove bold, you have to calculate the ADC, because by calculating the ADC, you divide the signal obtained at high B value, low B value, but the contribution of T2 and T2 star is the same. So you just remove the residual effect of T2 and T2 star by doing that. Now, I'm not sure I'm responding to your question, but there's an additional trick. It's a little bit technical, but I have to say it, I think. It has been pointed out that because of the Ball effect, the local gradients are modified by the propagating deoxyhemoglobin. So local gradients contribute to the B value. So the diffusion signal and the diffusion response we see might be a modulation of the local gradient and not something related to the tissue microstructure. It is it is true. So in order to remove this effect, what we need to do is to use a double spin echo sequence, double refocus spin echo sequence. By doing that, if the sequence is well designed, we remove sensitivity to local gradients. And that's what we have done in our 2006 PNAS paper and in the subsequent papers. We have been very careful not to use a plain spin echo sequence, so to avoid the critics that if we could see ball through the local modulation of the gradients. So I know it's complicated technically, but it is a pitfall that has to be avoided, right?
So is that the way you routinely do these at this point with this double echo experiment?
Yeah, most of, in clinical, in human, not always, because especially if we do the application, there's no point to do that. So if there is no ball confound, there's no need to do that.
Sure, that makes sense.
All right, so now if there are no other questions, so yeah, we can clap in our houses. Thank you so much, Denis, for this talk. It was amazing.
My pleasure. And again, you can put your slides on your website. So, because I was a little bit fast and I put some references, you can look at them and feel free to.
For the slices. And so also, we will probably make the video available in about a week or so. So thank you so much.
Thank you very much, Denis. And I think you and I are maybe scheduled to spend a few minutes together.
So
good.
Shall I switch to the other link or
switch to the other link? And I'll be with you in just a minute. But
okay.
Again, for everybody, on behalf of everybody, thank you for a fantastic talk and for taking the time to share with us.
And thanks a lot for inviting me. It was really a honor for me, and I'm just sorry I could not be physically present with you. It would be more fun, but anyway, later. Bye-bye.